The mechanism of the water-soluble hormones in a generic sense is the water-soluble hormones, again, cannot enter the cytoplasm because they are polar. They cannot go through the plasma membrane. They transmit the signal by binding to a receptor on the plasma membrane, and they transmit the signal through a second messenger mechanism. Please remember that the hormone is always the primary message, and it transduces the signal to a second messenger. And the second messenger can also then result in phosphorylation of enzymes to rapidly change activity. Protein phosphorylation of gene regulatory proteins to control gene expression takes place over several hours. So any pathway being turned on at the gene level takes hours. Any protein being turned on by protein phosphorylation can alter the activity of that protein within seconds or minutes. Some water-soluble hormones bind to receptors with intrinsic protein kinase activity, the tyrosine kinase class of receptors. And the best example of that is insulin. Other examples are growth hormones. They bind to a class of receptor called the tyrosine kinase class of receptor. In this figure are some of the major players involved in signal transduction by the water-soluble hormones. And on the left side of the table are the respective pathways, and most of these pathways have G proteins. The cyclic AMP pathway, which is a second messenger, results in the previous activation of a G protein called GS, stimulatory G protein. Depending on the receptor, another G protein can be activated called GI, inhibitory G protein. And when the G protein is either stimulated or inhibited, an enzyme is being affected, and that enzyme is called adenyl cyclase. And adenyl cyclase, that's the enzyme that makes cyclic AMP. And if cyclic AMP is being made, it activates protein kinase A. If cyclic AMP is not being made because the inhibitory G protein is being activated, adenyl cyclase will not work, cyclic AMP will not be made, and protein kinase A will not be stimulated. And examples of the hormones that use the cyclic AMP dependent pathway, major hormones, glucagon, epinephrine, and epinephrine can bind to different kinds of receptors, beta 1, 2, 3, alpha 1, alpha 2 receptors, and so on. And other hormones, vasopressin, located in the kidney. Another pathway that's going to be discussed in this chapter is the PIP2 pathway. There's a G protein present in that pathway called GQ, and that GQ affects a very important enzyme called phospholipase C. Second messengers involved there are diacylglycerol, IP3, as well as calcium. And the protein kinase being stimulated there is protein kinase C. Examples of hormones which use the PIP2 pathway are vasopressin as well as epinephrine when epinephrine binds to the alpha 1 adrenergic receptor. This is all going on in vascular smooth muscle. That will be discussed in this chapter. The cyclic GMP pathway, there is no G protein involvement, and there the key enzyme is guanylyl cyclase. And guanylyl cyclase makes the product called cyclic GMP, which is a second messenger. Once cyclic GMP is made, it activates protein kinase G. And what uses the cyclic GMP pathway is ANF, sometimes called AMP, for atrial natriuretic protein or peptide. If you see ANP or ANF, it's one and the same hormone. And that also uses cyclic GMP pathway is nitric oxide, all involved in vasodilation. Another signal transduction pathway catalyzed by a water-soluble hormone is insulin, as well as growth factors. A G protein is involved with insulin and growth factors, and that's a G protein called RAS protein. And RAS is also called P21, RAS protein, but it's a monomeric G protein. I'll talk more about what that means, monomeric versus trimeric G protein, coming up in this chapter. And downstream, what involved in the RAS pathway is a class of protein kinase called the tyrosine kinase class of receptor. Again, the hormones which use the tyrosine kinase class of receptor are insulin as well as growth factors, insulin-like growth factor, PDGF, epidermal growth factors. They use the tyrosine kinase class of receptor. Now, before we leave this figure, I want to stress here, of the second messengers, there is a second messenger, cyclic AMP, and cyclic AMP turns on protein kinase A. Another second messenger, cyclic GMP, and it turns on protein kinase G. Another class of second messengers, diacylglycerol, IP3, and calcium, they're involved in turning on protein kinase C. Notice here, don't get confused. What second messenger turns on which protein kinase? It's so confusing. But right here, as simple as I can state, which second messenger turns on which protein kinase? It's not difficult to remember this. All you need to know is cyclic AMP turns on protein kinase A. Cyclic GMP turns on protein kinase G. And calcium turns on protein kinase C. Notice the inflection of my verbiage to know what second messenger turns on which protein kinase. Now, regarding protein kinases, as well as a related reciprocal class of proteins called phosphatases, what protein kinases do, by definition, notice the suffix kinase. When you see the suffix kinase, that's telling you that is an enzyme that uses the terminal phosphate of ATP to stick that phosphate onto something. What's the something? You look at the first word. If the first word says protein, that's what the enzyme does. So a protein kinase, by definition, uses the terminal phosphate of ATP to stick that phosphate onto target proteins. By putting the phosphate onto a target protein, sometimes that protein is turned on, sometimes that protein is turned off. It all depends what protein is becoming phosphorylated, whether it's on or off. But that phosphate on the enzyme doesn't sit there forever. When another class of hormones like insulin or growth factors are being turned on, downstream they'll activate a class of enzymes called phosphatases. And what the phosphatases do, they remove the phosphate from those enzymes that were phosphorylated by protein kinases. And by removing those enzymes become dephosphorylated, and it depends what enzyme we're talking about, whether the enzyme, when it's dephosphorylated, whether it's turned on or off, whether the enzyme is turned on or off by the dephosphorylation by insulin or growth factor mediated activation of a phosphatase. So the alpha subunit in the presence of GTP is active, and if it's active, it then in turn activates adenocyclase on the inner leaflet of the plasma membrane of certain tissues. And then the adenocyclase in turn can make cyclic AMP to turn on protein kinases. But now the alpha subunit in the presence of GTP, it doesn't sit there forever as well. The GTP can hydrolyze down to GDP. So in other words, the alpha subunit has GDP ACE activity. The GTP has a certain half-life. It can be destroyed down to GDP. When it forms GDP, the alpha subunit can reassociate with the beta and gamma subunits, reforming the trimeric G protein, alpha, beta, and gamma, and GDP is present and it's inactivated, waiting for the receptor to become activated so that GDP can be displaced with GTP. Here is the first of 3 signal transduction pathways, and this first one is the signal transduction pathway that's mediated by a major class of hormones, glucagon and epinephrine, as well as vasopressin. On the upper left-hand side of the page, because glucagon and epinephrine are water-soluble hormones, they cannot enter the cell. Therefore, they have to bind to a class of receptors. And this class of receptors is visually shown as a 7 transmembrane domain protein, sometimes called a serpentine receptor because it spans the membranes multiple times. But because the hormone, either glucagon or epinephrine, cannot enter the cell, they bind to the serpentine receptor. And what kind of receptor and where is it located, that's the physiology part. There's different kinds of these receptors, beta receptors, beta 1, 2, and 3, whether it's cardiac or smooth muscle. That's all discussed in physiology, or alpha 2 receptor. But the important feature biochemically is that when the hormone binds to the receptor undergoes a conformational change and in turn activates the trimeric G protein called GS. And then alpha S of GS binds GTP by mechanisms we just discussed. Alpha S binds the GTP, beta and gamma go away. Alpha subunit now becomes activated. Alpha subunit in turn activates on the inner leaflet of the plasma membrane adenyl cyclase. And what adenyl cyclase is going to do is take a little bit of ATP in the cytoplasm, take off the 2 terminal phosphates, and cyclizes the remaining phosphate on the ribose to make 3', 5' cyclic AMP as the product, but more commonly called cyclic AMP as the product. Now before we move on, when epinephrine binds to the alpha 2 adrenergic receptor, alpha 2 adrenergic receptor turns on a trimeric G protein, not GS, but it's called GI, inhibitory G protein. Alpha I of GI binds GTP. Alpha I then inhibits adenyl cyclase, preventing adenyl cyclase from making cyclic AMP. So a stimulatory G protein turns on adenyl cyclase, inhibitory G protein inhibits adenyl cyclase from making cyclic AMP. Now once the cyclic AMP is made when GS is stimulated, cyclic AMP in turn activates protein kinase A. And protein kinase A, by definition, because it's a kinase, will use ATP to phosphorylate certain enzymes. On the lower right-hand part of the page, some enzymes become phosphorylated, and depending on what enzyme we're talking about, some enzymes are stimulated, other enzymes are inhibited by that phosphorylation mechanism that goes on. Now another item here is the cyclic AMP. The cyclic AMP, what happens to it is once it's made, it can do one of 2 things can happen to the cyclic AMP. One thing is it can activate protein kinase A, and the other thing can happen to the cyclic AMP is it can be destroyed. And it's destroyed by an enzyme called phosphodiesterase. And the phosphodiesterase destroys cyclic AMP to 5' AMP as the product. So one more time, 2 fates of the cyclic AMP. One is to activate protein kinase A to phosphorylate certain enzymes. Other fate of cyclic AMP is to be destroyed. On the upper right-hand part of the page, when vasopressin or epinephrine binds to the alpha 1 adrenergic receptor, when that receptor undergoes a conformational change and becomes stimulated, it in turn activates another G protein called GQ. And then alpha Q of GQ binds GTP when the receptor is stimulated. Alpha Q of GQ binds the GTP, beta and gamma go away. Alpha Q in turn activates yet another enzyme on the inner leaflet of the plasma membrane, not adenyl cyclase. This enzyme is called phospholipase C, and phospholipase C takes a lipid in the membrane called PIP2. PIP2 is cleaved right down the middle, generating 2 fragments. One fragment is called diacylglycerol, other fragment is called IP3. IP3 in turn then goes to the ER, and in the ER will then open up a channel for calcium. Calcium now will leak out of the channel into the cytoplasm, and calcium along with diacylglycerol together will stimulate another kinase called protein kinase C on the right-hand side of the page. Protein kinase C, because it's a kinase, will then stimulate the phosphorylation of certain enzymes. So some enzymes become phosphorylated, And depending on what the enzyme is, some enzymes are being turned on and some enzymes are being turned off by the phosphorylation mechanism that just went on. Now, if the National Board were to ask you, what are the second messengers for the alpha-1 adrenergic receptor system, you say diacylglycerol, IP3, and calcium. But if they were going to ask you, what are the direct activators of protein kinase C, you answer by saying diacylglycerol and calcium, not IP3. IP3 is an indirect activator because IP3, notice it's stimulating the secretion or the release of calcium out of the ER. And then calcium, along with diacylglycerol, stimulates protein kinase C. And then protein kinase C results in phosphorylation. And as you all know, this has a lot to do in physiology of increasing smooth muscle contraction. Now, coming back to this issue of cyclic AMP in the middle of the page. So the cyclic AMP can stimulate protein kinase A to cause phosphorylation of certain enzymes. Now, if you were asked what pathway might be stimulated here on the lower right-hand part of the page, well, you would think, well, what's going to be stimulated would be something like glycogen breakdown. Because after all, why are you secreting glucagon? Why are you secreting epinephrine? It's because the blood glucose is dropping. And if a blood glucose drops, you want to stimulate glycogen breakdown to generate glucose into the bloodstream. But glycogen will run out. And when glycogen runs out, the body has to have another pathway that's going to be stimulated to try to maintain blood glucose. And what's going to be stimulated is the pathway called gluconeogenesis, stimulating of that pathway. Now, how does that happen when you secrete glucagon? Well, that's the lower left-hand side of the page. So again, when glucagon binds to its receptor, it stimulates GS to stimulate adenocyclase to make cyclic AMP, which stimulates in turn protein kinase A. Now on the lower left side of the page, the protein kinase A, it can in turn phosphorylate another protein called CREB, which stands for cyclic AMP response element binding protein. And CREB is normally inactive. And by phosphorylating CREB protein, it's now activated. And CREB will go into the nucleus of the liver cell. That's where it's located, in the liver. And the CREB protein will go into the liver cell and go into the nucleus of the liver cells. And there it looks for a piece of DNA called the CRE, which stands for cyclic AMP response element. Look at the words. Listen to the words. Cyclic AMP response. Responding to the cyclic AMP. Cyclic AMP response element. It's a piece of DNA which is part of an enhancer region for a very important enzyme in gluconeogenesis. Don't worry about the enzyme right now. That's discussed later on in metabolism. But a major enzyme in gluconeogenesis is being stimulated at the gene level because the CREB protein binds to the enhancer. An important gene is stimulated or turned on. Gene expression is going to go on for gluconeogenesis. Now, if you want to know a little bit more detail of that, turning on of gene expression of gluconeogenesis, go back to page 78 of the Biochemistry lecture notes. That's in chapter 5. You will see how glucagon and the CREB protein involvement turn on gluconeogenesis. Please remember that the CREB protein is also classified as a specific transcription factor. A specific transcription factor, which is discussed in chapter 5, specifically on page 78. So notice here, to summarize this figure, when the blood glucose drops, we want to maintain blood glucose values in order to prevent death from occurring. And so how are we going to do this? On the lower right-hand part of the page, we're stimulating glycogen breakdown. On the lower left side of the page, we're stimulating gluconeogenesis, such that when glycogen is gone, and it will be gone, gluconeogenesis will be at its maximum, trying to maintain blood glucose values. Coming up next is the second of the three signal transduction pathways that undergoes mediation by water-soluble hormones. This one here is the cyclic GMP activated pathways. Now, there's several different ways to generate or stimulate cyclic GMP production. We all know this goes on to cause vasodilation. On this figure is the biochemistry. How does vasodilation occur when cyclic GMP is being made? Under conditions of distension in which ANF, or sometimes referred to as ANP, the atrial natriuretic peptide or factor, you might say it by either name on board exams, ANF or ANP, when they are being stimulated under conditions of distension, the hormone will bind to a receptor. The receptor is not an adrenergic class of receptor. It's not a serpentine receptor. It just spans the membrane one time. When the hormone binds to the receptor undergoes stimulation, and the receptor also is an enzyme. And it's an enzyme on the cytoplasmic side, will take a little bit of GTP in the cytoplasm, take off the two terminal phosphates, and makes it into cyclic GMP as a product. Specifically, it's called 3', 5' cyclic GMP, but just abbreviated cyclic GMP being made as a product. Now we'll come back to the cyclic GMP in just a minute. On the upper right-hand part of the page, under the conditions in which vasodilators are being secreted, the vasodilators will go to the vascular endothelial cells and stimulate a major enzyme called nitric oxide synthase. Nitric oxide synthase, as the name implies, will synthesize nitric oxide by using the substrate arginine. And arginine, no one expects to know the structure of arginine, but it has a lot of nitrogen in its R group. One of the nitrogens of arginine is taken off by nitric oxide synthase. That nitrogen is then converted to nitric oxide. On the upper right-hand part of the page, under conditions of certain pharmacological drugs, nitroprusside for the treatment of hypertension, nitroglycerin for the treatment of angina, isosorbide dinitrate for the condition of treating angina as well. Notice that these drugs have nitrogen in them. When they are administered biochemically, they are then converted to nitric oxide. So on the upper right-hand part of the page, notice the integration of physiology, pharmacology, as well as the biochemistry generating nitric oxide. Once nitric oxide is generated, it passes right through the membrane of the vascular smooth muscle cells. There is no receptor for nitric oxide. It's permeable to the membrane, comes right into the cytoplasm of the vascular smooth muscle cells. And there it sees an enzyme called guanylate cyclase. You might see it called guanyl cyclase without the ATE ending. But the important feature is this enzyme, just like adenyl cyclase on the previous page, guanyl cyclase will take a little bit of GTP in the cytoplasm, take off the two terminal phosphates, and make cyclic GMP as the product. Notice this cyclic GMP can either be made from ANF stimulation of its receptor or by nitric oxide stimulation. Once cyclic GMP is made, it activates protein kinase G, and because it's a kinase, after a series of steps that's discussed in pharmacology, protein kinase G will then result in vasodilation that goes on in the vascular smooth muscle cell. Another item about the cyclic GMP, just like the previous page, if they were to ask you what's the fate of cyclic GMP, you answer by saying its fate is to stimulate protein kinase G, but another fate of cyclic GMP is to be destroyed. It's destroyed by another phosphodiesterase in the smooth muscle cells called phosphodiesterase 5, and what the phosphodiesterase does, it destroys the cyclic GMP to 5' GMP, which cannot activate protein kinase G. The third of the signal transduction pathways to review is the signal transduction pathway that's mediated by insulin as well as growth factors. So insulin and growth factors have a similar mechanism with one another, but the location of the receptors are vastly different. The receptor, obviously, for insulin is differently located compared to the one for growth factors. But the mechanism, the biochemical mechanism, is somewhat similar. So the issue of insulin is what's going to be discussed on this particular figure. When insulin or a growth factor finds its receptor on a specific cell, two things happen to the receptor. The receptor dimerizes, and then the receptor becomes activated. And the receptor becomes activated, it has tyrosine kinase activity. Now, what does a tyrosine kinase activity mean? Because it's a kinase, by definition, it uses ATP, in this case here, ATP that's in the cytoplasm, and uses it to put the phosphate of ATP onto target tyrosines. But now, where are the target tyrosines? On itself. So it puts phosphates onto itself. It's called autophosphorylation. So one more time, in the presence of insulin, two things happen to the receptor. It dimerizes and it autophosphorylates itself. Now, once the receptor autophosphorylates, it becomes activated. And when it becomes activated, what happens is there are a series of proteins that can now bind, and one of these proteins is called IRS1, which stands for insulin receptor substrate. So this protein located in the cytoplasm will bind to the receptor for insulin only when the receptor phosphorylates. So the IRS1 docks, and then in turn another series of proteins dock with IRS1. These other proteins are classified as SH2 domain proteins, and one of them is called PI3 kinase. This protein, when it's activated, in turn results in the opening up of a vesicle for GLUT4 in adipocytes and muscle. Remember, two organs that have a receptor for insulin are muscle and adipocytes, and in these cells the vesicle for GLUT4 is opened in the presence of PI3 kinase, and GLUT4 is put onto the surface of these particular cells. Another SH2 protein can dock. The name of this one in the figure is not important to know for boards, but downstream there is a G protein called RAS, which now becomes activated and in turn activates specific transcription factors that cause certain genes to be turned on when RAS is activated. Now, RAS, it's a G protein, but in contrast to the other G proteins we talked about earlier, this RAS is a monomeric G protein. It is a single peptide in contrast to the three peptides that make up GS or GI. This is a monomeric G protein. Molecular weight is 21,000, P21. But it binds GTP in order to work. So if what's being turned on is insulin mediation, then what genes are being turned on by insulin? Genes involved in triglyceride synthesis, cholesterol synthesis, and so on. If the hormone is a growth factor, what genes are being turned on by RAS mediation are genes involved in cell division. So this idea of gene expression in the nucleus. What genes? It depends what hormone we're talking about and what organ or tissue for that particular hormone. What genes are being turned on. The last SH2 protein class that can bind, again, is not important. But when this SH2 protein class is turned on, not shown here with these arrows, on the lower left side of the page, there are other intermediate proteins becoming activated. They're not important for boards. In fact, they're still being researched as we speak. But the end product of all this activation of a cascade of proteins is a phosphatase. And this phosphatase is now activated in the presence of insulin or growth factors. But this case here of insulin, therefore, when the phosphatase is activated, will dephosphorylate these enzymes, and these enzymes now become dephosphorylated. And when the enzymes are dephosphorylated, some enzymes are turned on, and some enzymes are turned off by that dephosphorylation mechanism. Now remember, 2 figures ago, we talked about the idea and reviewed. When glucagon or epinephrine is secreted, epinephrine and glucagon activate a kinase, protein kinase A, which results in phosphorylation of certain enzymes. And notice on this figure how insulin results in the dephosphorylation, basically of the same enzymes that epinephrine and glucagon resulted in their phosphorylation. Now I want to illustrate how important this is. Phosphorylation, dephosphorylation on the next figure. On this figure is a great example of the reciprocal effects of glucagon and insulin. And we're going to talk about it in reference to glycogen metabolism. Now the details of glycogen metabolism are discussed later on in chapter 15, but right now we're in chapter 9. The idea is at the top of the figure, under conditions in which blood glucose drops, you secrete glucagon. Glucagon will result in the activation of protein kinase A. Now the details was discussed earlier in the chapter, how protein kinase A is turned on by glucagon. But once protein kinase A is stimulated, it uses a little bit of ATP in the cytoplasm, takes that ATP, and one of the phosphates of the ATP by protein kinase A is put onto an enzyme called glycogen phosphorylase. So the enzyme is now phosphorylated. Another phosphate is put onto glycogen synthase, and glycogen synthase is now phosphorylated. And it just so happens that glycogen phosphorylase with the phosphate group on is active. Glycogen synthase with the phosphate group on is inactive, such that glycogen is broken down and not made when you secrete glucagon. Exactly what you want to happen under conditions of fasting or starvation. You want to break down glycogen and not make it. But that phosphate doesn't sit there forever on these enzymes. Under conditions in which the glucose levels in the blood rise, you want to make glycogen, you don't want to break it down. So you secrete a hormone called insulin. Insulin will then activate a phosphatase. Again, the details of how insulin results in the activation of the phosphatase is discussed on the previous figure. But once the phosphatase is activated, the phosphatase will then dephosphorylate glycogen phosphorylase, dephosphorylate glycogen synthase, and it just so happens that dephosphorylated glycogen phosphorylase is inactive, dephosphorylated glycogen synthase is active. Consequently, you make glycogen, but don't break it down at the same time. So it's beautiful how this works. Reciprocal phosphorylation, dephosphorylation that goes on.